So it's been very exciting to see how radiomics has grown as a field and how the application, not only of the new technology that's being developed, but it's also the application of how we're thinking through what this might mean in terms of the world that I live in, which is oncology and clinical trials. And there's really a lot of different ways that we see radiomics being useful. I think that, in the beginning, we were just trying to do the old stuff better. So what I mean by that is using, say, like the RESIST criteria. Is there just a better way for us to do RESIST with radiomics? And of course, it continues to evolve. And now we think, and we'll talk more later, hopefully, about what are some of the new potential uses in clinical trials to get to what we're all trying to do, which is better outcomes for patients. So better treatments for individual patients, more personalized care. And then working through to maybe one day be able to use radiomics and other features to help us think about prevention or just treating the patients that really need to be treated, but getting to them earlier.
And again, I think you make a really interesting point there in terms of the range here of possibility, because obviously one of the things that, images are really a hallmark of the milestones of the patient's journey in many instances. And so we can see this really the applications from early identification and screening through treatment and evaluation of the treatment success and so forth. So again, just a great opportunity here for an expansive set of use cases. So Fabian, perhaps you could share your perspective as a practicing physician who's hard at work in trying not only to develop the research, but also find ways in which to apply the capability.
Yeah, thank you, Rose. It's a pleasure to be here, and thank you so much for the invitation. As you pointed out, I'm in the middle of a busy day, so hopefully I don't get bothered too much by pages. My experience with radiomics as a pulmonologist and as an interventional pulmonologist in particular started out probably 12, 13 years ago when I was a fellow at Mayo. One of my projects was to look at characterization of lung tissue in idiopathic pulmonary fibrosis, so not lung cancer, with a quantitative imaging tool that had just been developed at Mayo. And the problem about idiopathic pulmonary fibrosis at the time was very similar to an emerging problem, which was that of characterizing the different tissue types within lung adenocarcinoma. And there was the reclassification of lung adenocarcinoma that came out at the time, showing a great correlation between quantitative histology characterization and then patient outcomes. And so the question was, could we replicate this problem with what at the time we did not call radiomics, that came later on, but with the images that we had. And so we had the engineers at Mayo adapt this ILD predicting model to focal lesions, and that's how I got started in radiomic research. And this was a fascinating project that led on to other projects looking at benign versus malignant. So my work as an interventional pulmonologist, of course, is at the very forefront of patient management. We get patients with lung nodules, indeterminate pulmonary nodules, and we have to decide whether they're malignant or not. If they're malignant, what kind they are. And so certainly biopsy is what I do for a living, so I hope that doesn't go away. But there's a lot of this information that we really can get from sophisticated radiomic algorithms. And so that's very interesting to us.
And I think, one of the last points you made relative to the biopsy opportunity here, it really can be somewhat of a non-invasive biopsy or a proxy for that physical examination, which certainly when you're working in very sensitive areas like the brain, for example, anything you can do to reduce the need to actually go in and disturb the area is certainly to the benefit of the patient. And so, again, great thoughtfulness there in terms of the comments.